Okay, good luck. Maganda naman yung thesis. Thank you pala dun sa PDF ng presentation.
Ay, yes sir.
Okay. Galing. Parang kuwan nga eh. Parang ano ba to? May napanood akong TV episode na ganito eh.
Dexter po ba yan, sir?
Yeah, I think, yeah.
Yeah.
Nagtatrabaho siya sa forensics. Ganun.
Yeah, opo.
Sige, good luck.
So, hindi ko pa napapanood yun.
Hindi mo
Sige
pa
po, sir,
napapanood?
thank you. Old na ata yun, sir, na ano eh, palabas.
Oo, matagal na. Nung nagkaanak ako, hindi na ako nanonood ng TV.
Dexter po ba yan, sir?
Yeah, Dexter.
Yeah.
Nagtatrabaho siya sa forensics.
May delay ang live streaming, ano?
Yes, ma'am.
Malaki yung delay. Okay.
Okay. Ay, nakikita pala tayo, ma'am, dun sa pag sinare ko na po yung screen ko, ma'am, dun sa dito sa Zoom, ang makikita na lang dun sa live stream ay yung presentation na lang, ma'am, no?
Yes, at I think kasama yung face mo.
Yeah, i-try mo i-share yung title page. Para makita.
Oh, pwede yun.
Screen, yes. Share F5. Ayan.
May delay lang ha, medyo malaki yung delay nung YouTube. Punta ka dun sa okay, try na nating i-set up yung katulad nung dati. Yung i-ready mo na yung presentation mo. Yan.
Kita po ba, ma'am?
Yes, large siya. Very
okay.
Good. So, may latency yung YouTube, pero okay lang. At least nakikita na. I think mga 10 or 20 second delay.
Kita rin po, ma'am, yung laser pointer, no?
Oo, kita yan.
Yeah. Okay. Medyo nadi-delay po kasi dito sa akin.
Sige, hintayin na lang natin yung panel mo. Remind natin. Okay, yan na. Sino to? Si K. Bernandez.
Girlfriend ko po, ma'am.
Oh, hello, girlfriend. Okay. Check natin nag-confirm naman din si Dr. Paraan. Tama?
Opo. Ang hindi lang po nag-reply dun sa email ay si Dr. Hana.
Wala akong email. Cellphone number niya. Nakaka-chat mo ba siya?
Ay, hindi po, ma'am. Through email lang po yung
Sige, hintayin lang natin sila. Yan na si Dr. Bardalosa. Si Dr. Paraan na lang.
Good afternoon po, ma'am. Connect up. Connecting. Wala.
Good afternoon, Dr. Bardalosa. Co-host ka, no, Wilbur? Tama?
Opo.
Okay. Bakit guest ang nakasulat dito sa account mo?
Ay, hindi po ba?
Allow make co-host. Di bale, gagawin kitang co-host ngayon.
Hindi po, ma'am.
Okay. Alright, isa na lang. Lalarga na.
Good afternoon, Dr. Hana.
Afternoon, hello po.
Ito rin kayo sa NIP, ma'am.
Yes, Ma'am Jing, nandito po ako sa Tento.
Na-receive niyo po ba yung sinend kong PDF copy ng slides? Nag-email po ko kaninang siguro around 12 or 11:30, I think.
Ngayon ko lang nakita.
For your reference lang po, ma'am.
Si Dr. Paraan na lang. Ritz, pwede bang gawin kitang co-host din para paniguro?
Ngayon ko lang.
Thank you, Ritz.
For your reference lang po, ma'am. Ay, nahuli.
Okay, si Dr. Paraan na lang. Host din. Chat ko na si Dr. Paraan. Wala pa siya eh. Hindi niya daw makita yung yung URL. Okay. Sandali ha. 8957. Yan na. Okay.
Ayan.
Yan na. Kompleto na tayo.
Ayan.
Okay.
Afternoon, Dr. Paraan.
Okay, I'll start recording. How is your connection, Dr. Paraan? Oh, I'll be on audio. Thanks. Okay. And usually, ang reader ang nag-chair. Okay. Sige, di bale. I'll chair instead. At least to start. And then I'll ask one of the examiners to chair. Okay. So, good afternoon, everyone. Yes. Okay, examiner will chair. So paperwork, yes. So I will introduce, but may I ask Dr. Ana Bardolasa to be the chair? Is it all right? Okay. So I think 45 minutes discussion, presentation, 45 minutes discussion. So Dr. Bardolasa, would you introduce the... Hindi, ako na lang mag-introduce, tapos si Dr. Bardolasa will chair the question and answer. Is that all right for everyone?
Yes, ma'am, that's okay.
Introduction lang. So, good afternoon, everyone, to the thesis defense of my master student, Wilbur Galarion. The panel members are myself as the advisor. The reader is Dr. Francis Paraan. The examiners are Dr. Armando Somintak and Dr. Hannah Bardolasa. The chair of the examination panel will be Dr. Hannah Bardolasa. Wilbur will be given 45 minutes for the presentation and 45 minutes to an hour for the examination. Thank you. So Wilbur, if you're ready, you may start.
All right. Thank you, ma'am. So again, welcome and thank you everyone for attending the defense of my master's thesis research entitled Transient and Steady State Blood Droplet Stain Patterns on Fabric with Backing Material. Again, my name is Wilbur H. Galarion Jr. I'm from the Video and Image Processing Laboratory, and my advisor is Dr. Maricor and Soriano. So we begin our discussion by defining forensic science, which is an application of science in the field of law, particularly in criminal law. In criminal investigation, perhaps the most important part is evidence gathering. Forensic investigators use several forensic tools in analyzing crime scenes and processing evidences. Forensic tools such as DNA analysis, fingerprint, DNA testing, fingerprinting, ballistics, among others. In violent crime scenes, perhaps the most commonly found evidence is blood. Bloodstain evidences can be found almost anywhere deposited on various surfaces. Bloodstain pattern analysis, or BPA, is a forensic tool used in analyzing these bloodstain evidences. BPA is an interesting field of research for forensic scientists because it provides an objective method based on empirical and physical models to reconstruct crime scenes. In the Philippine scenario, forensic science is less used in criminal investigation. In fact, up to 90% of cases rely solely on the witnesses' testimonies. This is according to the study of Go and de Ungria in 2019. The reason for this is there is a general lack of resources, facilities, and qualified personnel in the field of forensic science. Hence, research such as this can potentially be very useful. So what then is bloodstain pattern analysis? Well, the goal of bloodstain pattern analysis is to reconstruct crime by examining the characteristics of the bloodstain patterns. Characteristics meaning measuring the size, shape, and distribution. In doing so, relevant information can be obtained that can be used to determine the events that transpired in a crime scene. One of the main techniques used in BPA is trajectory reconstruction of individual stains. By relating the stain size to the impact conditions, information such as impact velocity, directional angle, impact angle, and possibly the volume of the droplet that produced these stains can be obtained, which can be used to solve the equations of motion that can reconstruct the projectile path of the droplet. This gives us an insight in the position of the victim, the position of the culprit or the perpetrator. This can also give us insights on whether the victim is sitting, laying down, or standing up. Bloodstain pattern analysis can also give us information on the bloodletting event. By investigating the distributions of bloodstains and examining the global features, different staining events can be obtained, such as stains resulting from the swinging of objects or stains resulting from objects hitting a pool of blood. There have been several researches on bloodstain pattern analysis, most common of which are on BPA bloodstain patterns on hard non-wetting surfaces, most of which are on trajectory reconstruction. The simplest method is the stringing method, where the trajectory of the bloodstains are approximated as straight lines. Of course, this presents considerable inaccuracies, and hence more accurate methods were studied. In particular, one that includes air drag and gravity effects, as well as wind currents, to produce a more accurate reconstruction of the trajectory. There have been research on trajectory reconstruction as well as an optimization problem. Now, the behavior of the droplet immediately after impact has also been extensively studied. In particular, the drop size and the formation of structures along the rim of the droplet, as well as the maximum diameter at maximum spreading and its relation to the impact velocity are also investigated. Less researched are bloodstains on fabric surfaces. Most of this research focus on the wetting and wicking behavior of the droplet to understand the dynamics of how bloodstains form in fabric. The focus is also on the parent stain characteristics, where only a descriptive analysis of the stain appearances was offered, and hence no clear empirical relationship between the stain size and the impact condition is presented. Now, the main reason why most of the BPA studies are on hard non-wetting surfaces is because the appearances of the stain do not change over time, and hence the impact conditions of blood stains are easily retrievable. This is not the case for blood stains on fabric. Because of the absorbent nature of fabric, the stain shape and sizes change over time due to imbibition, as you can see here on the animation. Imbibition is the wetting of the fluid into the fabric and subsequent wicking. And so hence relevant information that can be used to reconstruct the trajectory may be lost. That is why it is significant to study fabric as a deposition surface. In particular, blood evidences are not found only on hard non-absorbent surfaces, but also on fabric materials. Backing material is also important to study because in crime scenes, most of the fabrics have materials underneath them. Examples of which are sofa covers, bed sheet, clothing, articles of clothing worn over skin. It's also important to be able to establish a relationship between the bloodstain sizes to that of the impact condition, particularly the impact velocity, something that has been only attempted on hard non-absorbent surfaces. And so this study will investigate both the transient behavior and the steady state characteristics of the droplet stains on fabric. You see the image here of the evolution of the droplet stains on fabric, and we can subdivide that into 5 different stages. The first stage is the initial droplet impact up to the maximum extension of the droplet. At the maximum extension of the droplet, you see here the characteristic formation of finger-like structures. Immediately after the maximum extension, rebounding occurs where the fingers are retracted up to this stage. After the retraction, initial wetting can be observed and subsequent wicking will happen up to the drying of the blood stains. Now, the objective of this study is to answer these questions, particularly in the transient behavior. We would like to look at the maximum extension and answer these 2 questions. Does the backing material have an effect on the spreading behavior of the droplet? And 2, looking at the formation of the fingers, will introduction of a backing material have an effect on the formation of the fingers? In the steady state characteristics, we aim to answer these 2 questions. Again, does the backing material have an effect on the parent and satellite stain characteristics? And can the impact condition, particularly the impact velocity, be retrieved from the blood stains on fabric? So we first look at the transient behavior of droplets. And this is done by investigating the maximum spreading ratio of the droplet immediately after it hits the fabric surface. The maximum spreading ratio is just the ratio of the maximum diameter of the droplet at maximum extension over the initial droplet volume, initial droplet diameter. So this one. Before we proceed further, let me just define some dimensionless numbers that will be used in this section. The first is the Reynolds number, which is the ratio of the inertial forces to that of the viscous forces. Mu here is the viscosity of the fluid. In this case, it's the viscosity of blood. And the second one is the Weber number, which is the ratio of the inertial forces to that of the capillary forces. Sigma here is the surface tension. And so a model for the maximum spreading ratio is derived via the energy conservation approach. Pasandide, Fard, et al. Considered the energy of the droplet, the kinetic energy of the droplet, its surface energies, as well as the viscous dissipation. The expressions for the viscous dissipation are given here, and the surface energies. Tc here is the time it takes to reach maximum extension. Omega here is the volume of the fluid, and phi here is the viscous dissipation function. And so we solve this equation and we get the equation for beta max, which is the maximum spreading ratio. Again, it's the ratio of the maximum diameter over the initial droplet diameter. And we get this expression here. Now, if we ignore the capillary effects, which is using this approximation, then we get a simpler expression given by this equation right here, where beta max is proportional to the 4th root of the Reynolds number. There are, of course, other models that other models that are used in the maximum spreading ratio, particularly these three, where the beta max is proportional to the fifth root of the Reynolds number. This is obtained when only surface energies are considered. Scaling as the square root of the Weber number is obtained when the capillary forces are considered, and scaling as the fourth root of the Weber number is obtained when conservation of momentum is considered. Sorry. So in the experiment, we use a passive drop experimental setup where we have a micro droplet injector placed directly above a polycotton fabric. A backing material is placed underneath the fabric, where we used leather and aluminum as the backing materials. The bulk modulus of the backing materials are given here. And the experiment is captured using a GoPro camera at 240 frames per second. The height of the microdroplet injector is varied to simulate different droplet velocities ranging from 0.4 meters per second to 4.4 meters per second. I should also note as well that the fabric has no tilting, so that only vertical drops are considered throughout the experiment. In this experiment, we use animals' blood, particularly swine or pig, with its physical properties given in this table. We use pig's blood because the properties of it is similar to that of human blood, which is given here. And so before we get the maximum spreading ratio, the raw images are processed first by doing a geometric transformation to correct errors arising from projections before binarization of the image is done. Now, a maximum inscribed circle, indicated by the red figure here, is fitted into the binarized image, where the diameter of the maximum inscribed circle will be our D max. So again, of course, D naught is the initial droplet diameter. And so we plot the maximum spreading ratio beta max against the Weber number for increasing Weber number, and we see that the beta max scales poorly with the two models presented earlier, particularly if we use conservation of momentum and when considering capillary forces. A better fit is observed when beta max is plotted against the Reynolds number, one when considering the spreading behavior dictated by energy conservation with viscous dissipation, which is given by this model right here, where the beta is proportional to the fourth root of the Reynolds number. You can see here as well that there is an overlapping of all the data points, fabric with no backing material, indicated by the black data points, and fabric with backing material, both leather and aluminum. And hence, you can say that the beta max may not be affected by the backing material. You see here that there is no significant change in the ratio if we introduce a backing material. The second part of the transient behavior is investigating the formation of finger-like structures along the periphery of the droplet. And to do this, we first consider fluid instabilities, particularly one that arises when two fluids are superposed one over the other, or when accelerated towards each other. The derivation of the dispersion relation is done by looking at the configuration here on the left side, where fluid 2 is superposed over fluid 1. And both fluid 2 and fluid 1 are under the effects of a gravitational field pointing downward. The interface is at z equals 0. Now, if we introduce small perturbations at the interface, the instabilities manifest itself as waves, as you can see here. When the fluid the density of fluid 1, rho 1, is greater than the density of fluid 2, we say that the system has a stable solution. This is the normal case when a lighter fluid is floating over a heavier fluid, and the instabilities only oscillate at the interface. Now, when the other case is when the density of fluid 2 is greater than the density of fluid 1, you have unstable solutions, and the instabilities grow exponentially, as you can see here. So we start with the Navier-Stokes equation, given by this equation, assuming a potential flow, where the assumed solution for the potential function is given by these expressions. Phi 1 and phi 2 here are the instabilities, or rather the perturbations at the interface, introduced at the interface. And so the dispersion relation is given in this equation, where it relates the angular frequency to that of the wave number k. Now, this theory was used by Richard Allen in 1975 to explain droplet splashing, particularly for unstable solution, that is when your rho 2 has a heavier or has a higher density than rho 1. And we can obtain a mode of maximum instability where k has a value of this given by this expression. Note now that the g on the previous equation for the dispersion relation is now replaced by a, since in a spreading droplet, the instabilities are now driven by the acceleration of the spreading droplet, not gravity. So we use a here instead of g. And of course, rho 2 is the density of the fluid. In this case, it's blood. And rho 1 is the density of your air. And we, from the wave number, we can then compute the wavelength of the interfacial waves, or the waves occurring at the periphery of the droplet, as you can see here, given by this expression. Note now that your rho 1 is, since the density of air is smaller than the density of blood, we can then ignore that, so that only rho here depicts the density of your blood. And so from this, we can then compute the number of waves occurring at the periphery of the droplet. This is done by dividing the circumference of the droplet at maximum extension, which is pi d, divided by the wavelength. And so we get an expression for the number of waves, or essentially the number of fingers at the periphery, to be this expression. So n is proportional to the square root of your Weber number. And so an empirical model that fits better with experimental data was obtained by Hall-Smith et al., given by this expression. So this equation fits better with experimental data, where Cn here is an empirical correction factor. So to observe the formation of fingers in the experiment, we obtain a radius profile of each of the droplet images. This is done by taking each edge point of the binarized image and measuring its distance from the center of the maximum inscribed circle. Then we subtract the radius of the maximum inscribed circle so that we get an expression for r given by this. This is the distance of the edge point to the edge of the maximum inscribed circle. So this one. And we do this for all the edge points scanning in the azimuthal direction. And we plot it so that we can get a radius profile from 0 to 360 degrees. Now, a moving average was also done on the radius profile to avoid spurious detection of peaks arising from noisy pixels during binarization. Now, of course, theoretically, the formation of fingers will result in equally spaced and have equal widths of fingers. This is, of course, not observed in experiment due to the surface roughness of the fabric, which promotes deviations in the formation of fingers. So there may be merging and splitting of fingers. That's why you can see uneven distribution of the fingers. And so to further avoid spurious detection of the peaks, we set a criteria for the detection of peaks where the mean peak width, I should note as well that the peaks in the radius profile corresponds to the formation, to the fingers formed. And so the criteria is that the minimum peak width should be 0.3 and the minimum peak prominence is 0.9. The prominence is just the height of the peak in relation to a relative base. Now, these values here are determined via or were obtained empirically by observing the minimum or the smallest and the largest fingers formed in the experiment, observed in the experiment. So we plot the number of fingers against the Weber number, and we see a clear separation of the data between the fabric without backing material and fabric with backing material, both for leather and aluminum. The data without backing material follows the Rayleigh-Taylor instability prediction, where n is proportional to the square root of Weber number, so almost close, 0.46. And empirically, we show that the backing material is affects the formation of fingers, where the number of fingers now is proportional to the fourth root of the Weber number. As you can observe here, there are data points with relatively larger error bars. This may be due to the these are occurring at the lower velocities, and this may be due to the fact that the undulations or the fingers formed at lower velocities are not well defined because their wavelengths are larger, as you can see in these regions here, as well as in these regions, here as well, and in these regions right here. And so higher errors are produced for these data points for lower velocities. Again, we have shown here that there is a clear separation between the fabric with no backing material and fabric with backing material. In particular, when introducing a backing material, the number of fingers approximately doubles. However, there is no clear, it is not clear whether the hardness of the backing material affects the formation of fingers. You can see here that the bulk modulus for both backing materials have a difference of an order of 2 magnitudes. But as you can see from the data here, the results are almost equal. We also show here the characteristics of the fingers, particularly the average width of each finger. And the fabric without backing material produces larger wavelengths, indicated by a larger average width. And so less defined fingers are observed for this data as compared to the other two. You can also see here that for increasing impact velocity, which is at higher Weber numbers, the number of fingers appear to saturate or tends to a constant value, as you can see from the asymptotic value of the average finger width, or it approaches a constant value, which means that the length occupied by a single finger in the circumference of the droplet approaches a limit value for higher impact velocities. For the second part, we looked at this research looks at the steady state characteristics of the droplet stains. A similar experimental setup was used, given here. And 4 different backing materials were used. The stained fabric samples are dried in ambient conditions for 24 hours before examining the characteristics of both the parent stain and the satellite stains. The parent stain is the main stain produced by the droplet, which is shown here, the larger stain here. And the satellite stains are produced by the ejection of smaller droplets that are ejected from the main stain. The backing materials were chosen so that their corresponding bulk modulus differ by an order of magnitude, as you can see here, from 0.30 gigapascal to 380. So we first look at the parent stain characteristics, and the plot of the average parent stain area with impact velocity shows that there seems to be no correlation with the size of the parent stain with that of the impact velocity. There is no, as you can see, that there is no defined increasing or decreasing trend. However, if we look at the parent stain images, as indicated by this figure here, there seems to be a difference in the perceived color of the parent stain images. This is investigated further by converting the RGB images of the parent stain to LAB space and taking the average value of the L channel and plotted it against increasing impact velocity. And as you can see here, there is a noticeable increase in the mean L value as the impact velocity is increased. This result is most evident for the silicon carbide backing material, which incidentally is the most porous backing material. And so this study proposes a hypothesis to this observation, in that the lightness of the stain images may indicate the amount of blood on the fabric, and that the porosity of the backing material decreases the blood in the fabric. The idea is that for higher kinetic energies of the droplet, which is obtained when you have higher impact velocities, droplet velocities, allows the blood to penetrate through the thickness of the fabric and therefore interact with the backing material underneath it. Now, if the backing material is porous, the blood will seep through and decrease the amount of the blood in the fabric available for wetting. This can also be observed on the previous plot of the average parent stain area, where the lowest mean parent stain area is observed for the silicon carbide material. So this is the material used, the silicon carbide material used. And the highest average parent stain area is observed for aluminum, which incidentally is non-porous. So this is the material used. We look at the satellite stain characteristics as well. And we see that there is an increase in the mean number of satellites and a decrease in the mean area of the satellites as the impact velocity increases. However, there is an overlapping of the data for the different backing materials for leather, polystyrene, aluminum, and silicon carbide. So the backing material may not have an effect on the formation of the satellite stains. Looking at the distribution of the satellite stains, by measuring the distance of each satellite stain from the parent stain, which is D, and we get the mean distance, plotted it against the impact velocity. And we see that satellite stains, from this figure, we see that satellite stains do not necessarily get ejected at farther distances when you have a higher velocity. It was also observed that there is greater variability in the location of the satellite stains for higher impact velocities, as observed by an increase in the error bars for higher impact velocities, comparing it to lower impact velocities. It's also observed that most of the satellite stains, about 60 to 80%, are located in the vicinity of the parent stain. This is the region between 0 to 60 millimeter from the parent stain. And about 50% of the total population of the satellite stains are located very near the parent stain, which is at the region between 0 to 20 millimeters. This is observed for all impact velocities. Hence, since there seems to be no dependence on the packing material used for the satellite stain characteristics, we then looked at the dependence of the satellite stain characteristics to the initial droplet volume. And clearly, right away, we see that there is a noticeable decrease in the number of satellites as the volume of the droplet is decreased. As you can see when you scan the image on the left from left to right. This clear decrease in number can also be seen in the plot here, where there is a clear separation of the data for the different droplet volumes. An empirical model was fitted to the data where the number of satellites is proportional to the square of the impact velocity, and A is a parameter that is linearly proportional to the droplet volume, as you can see here. And this result is consistent for all data of different packing materials. Now, this result gives us an idea that there is a possible method for predicting or calculating the impact velocity from stains on fabric. This is done by counting the number of satellites produced by the impacting droplet. Finally, we looked at the sizes of the satellites, and we see that there is a decrease in the mean area of the satellite as the impact velocity increases, so a decrease. However, at higher velocities, this decrease becomes less evident, as you can see here. So the change in the satellite area is less evident. If we look at the rate of change of the area at increasing impact velocities, we see initially a relatively large change in the delta A. However, this decreases at higher impact velocities, hovering around the 0 value. And so while this result does not give us a method of predicting the impact velocities or a clear way of calculating the impact velocities, this result can give us an insight that for higher velocity impacts, small satellite stains are observed which vary little in sizes. And for lower impact velocities, larger satellite stains can be observed that vary depending on the impact velocity of the droplet. So to summarize the results of this research, again, we have investigated the transient behavior and the steady-state characteristics of blood stains on fabric. In particular, we would like to answer the following questions presented earlier. So the first question is given here, where we show that the maximum spreading ratio is not affected by the backing material, and that the mechanism for the initial spreading of the droplet is energy conservation with viscous dissipation. The formation of fingers at the periphery of the droplet is influenced by the backing material, where we see that when a backing material is introduced, the number of fingers approximately doubles. In the steady-state characteristics, we see that we have shown that the parent stain characteristics showed a correlation with the porosity of the packing material, and that the satellite stains are not dependent on the backing material, but on the initial droplet volume. And we have shown here as well that there is a possible way of retrieving the droplet velocity. This is done by looking at the number of satellite stains produced. And so this result gives us an insight. The results of this research gives us an insight in the application of forensic investigation. Firstly is when considering materials underneath a bloodstain fabric evidence. For measurement of droplet fingers, backing material should be considered as shown in the results earlier. And for dried stains, particularly in satellite stain measurement, backing material can be ignored or can be excluded. If this method is to be implemented in crime scene investigation, measurement of satellite stains can be done in the vicinity of the parent stain where most of the satellite stains are located. The satellite stain sizes can give us an information or can distinguish between low and high impact velocity stains. And for trajectory reconstruction, where the impact velocity is needed, satellite count may be able to obtain this information from the droplet stains. Lastly, we look at some possible extensions to this study. First is by considering not only vertical but angle droplet impacts, both the magnitude and direction of the impact velocity, considering both the magnitude and direction. A preliminary result is presented here where we see a characteristic narrowing of the distribution of the satellites in the polar histogram as you increase the impact angle. So you can see a characteristic change. The second one is by looking at possible oscillations of the fabric during droplet impact and see if there is a damping effect introduced when a backing material is placed underneath a fabric. Lastly is by considering not just one but multiple droplets and be able to distinguish which satellite stain belongs to which parent stain. A possible method for this is an optimization approach. However, a fitting function must be formulated first. And so that is all for my presentation. Thank you for listening.
Thank you, Mr. Galarion, for your presentation. You have finished around 6 minutes ahead. So maybe we can proceed with the question and answer. Dr. Samantha, Dr. Paraan.
Dr. Paraan wrote a question. Is it okay, Dr. Paraan, if I read your question now? Dr. Paraan's question was it possible to do Fourier analysis or fingerprinting on finger radial profile that is major Fourier frequency proportional to 1 over the number of fingers? I will repeat. Was it possible to do a Fourier analysis on the finger radial profile that is major Fourier frequency proportional to 1 over the number of fingers?
Okay, so the idea of the finger formation, theoretically, of course, is for the, and so the idea is for the fingers to be formed equally spaced and will have equal width. So periodic, kumbaga, yung formation of fingers. However, in the experiment, because of the surface roughness of the fabric, this promotes merging and splitting of the satellite stains. And so I think Fourier analysis would And so Fourier analysis would actually give an inaccurate result, I would say.
Dr. Paraan chatted, I would think the opposite. Why do you think the Fourier analysis would give an inaccurate result? Fourier analysis may be more robust against artifacts. So what do you recall about Fourier series? Any signal can be expressed as? Do you remember your Fourier series, Wilbur? The Fourier series says any signal can be expressed as a linear superposition of sine waves.
I'm sorry.
Okay. The Fourier spectrum has information not readily visible to the naked eye, sabi ni Dr. Paraan. So if you have a sine wave, it is periodic. Okay. So if you try fitting a sinusoid in this up and down signals here, okay, then it will try to fit. So magkakaroon ka ng, you could get the sine wave that would be best fit to this up and down signals here. Okay. All right. So essentially the suggestion of Dr. Paraan is, or the question was, is it possible to do a Fourier analysis on the finger radial profile? Una muna, is it a yes or a no?
Well, so it can be, well, according to that, it can be used, particularly in trying to determine the peaks in the radius profile of the blood droplet.
Or even the best fit sinusoid. Okay.
Yes.
Dr. Paraan, so it essentially is a suggestion in a way. So, okay, Dr. Paraan,
Yeah.
yes, kasi your theoretical model involved waves. Okay, sabi niya. So indeed, if these are waves due to the instability, then dapat technically you can fit a sinusoid. Okay. Okay. So yeah, it can be, perhaps you can try and we can try and fit a sinusoid here in this radius profile of the blood droplet. Okay. All right. All right. Dr. Paraan, can I continue with your second question or suggestion? Or would you like to grill Wilbur some more? Sabi niya, yes, please. Okay, second question from Dr. Paraan. Or actually, this is a suggestion, a comment. Okay, so for future studies, perhaps the eccentricity of the stain will give details on the velocity impact angle. Can you comment on that?
Yes, it does give us the impact angle of the droplet. However, as I've mentioned in the introduction, the problem with studying blood stains on fabric is that the stains evolve through time, and so the size and shape changes. So the eccentricity might not be the same as when it initially impacted the surface, which is not the case for hard surfaces, not non-absorbent surfaces. So they retain their shape, and therefore the eccentricity can be calculated readily using the method mentioned earlier in the introduction, which is the Lmax over Wmax over Lmax. So it does give us the information on the impact angle.
So are you satisfied with the answer, Dr. Paraan? Thank you, by the way. Okay.
That is why also, can I just add, ma'am? That's why also in the outlook, the future experiments, the effect of the impact angle is also should also be studied so that we can see if the distribution of the satellite stain can give us that information. So not relying on the eccentricity, but on the distribution of the satellite stains.
Okay. Okay, Dr. Paraan said in the chat, thank you, by the way, it's a very good talk, very clear, informative, very easy to listen to. Okay. Thank you, Dr. Paraan.
Jing, ako naman.
Ay, si Ma'am Hana ang chair.
Ay, Hana, Doc Hana.
Yes, sir.
Tatanong ako kay Wilbur.
Apo, sir.
Okay. Wilbur, inassume natin dito na yung drops are actually spheres. Tama ba?
Yes, sir.
So
yes, sir.
uniformly distributed mass na sphere.
Yes. Yes po.
Kasi maganda rin ilagay dun sa outlook, I think, yung variation nung shape or practically the distribution of mass nung droplet as it falls, kasi magbabago yung shape niya just before it impacts.
Yeah, opo. Yes.
So magbabago yun.
So,
alam naman natin yun kasi meron siyang
yes.
may effect yung density ng air, basically, di ba?
Yes.
And then maganda yung itong slide na to, natuwa ako diyan kasi this is actually can be modeled by collision between the front end of the droplet and the later end, the tail end, just before they hit. So yung leading edge or leading side nung droplet tumama na, magba-bounce back, tatama siya doon sa incoming na tail end, at yung dalawang yon would actually cause ripples. Siya yung arm kung bakit arm, sorry, ano bang tawag natin? Fingers. Sorry, yung fingers or yung mga ganon, yung pagkakaroon ng star formation. Kasi pag nakuha natin yon, makikita mo na depende dun sa volume or size or the distance between the tail end and the front end, makikita mo kung magbabago yon depende sa volume, of course. And then makikita mo na magkakaroon ng relationship yung mga droplets. Okay? I mean yung finger formation. And Wilbur, yung image na tinitingnan natin dito, yung raw image, that's actually taken just after collision, just after impact, tama ba ako?
Yes po, at maximum extension of the droplet.
So yung sinasabi nating extension is actually still not, kumbaga hindi pa nag-land, tama ba ako? Kasi yung kamay nito parang nakahanap pa yung mga fingers.
Well, the droplet has already hit the surface, sir, and
yung fingers,
the fingers
hindi pa.
Form as if parang corona yung formation niya.
So Yeah,
So, papataas.
taas
Yeah.
pa siya. Yes, yes. Okay. Kasi I encounter something similar din sa bato na drain up mo sa tubig. Yung number of yung bounce back na
Yeah, opo.
ang ganda. Dito lang sa'yo, ang kinonsider mo yung fabric natin is flat. Flat, and then hindi katulad nung liquid dropping on liquid na magkakaroon ka ng recoil effect. Dito
Yes, opo.
minimal yung recoil pero obvious na mayroon dahil mayroon kang finger.
Yes. Yeah.
Tapos sisegundahan ko lang yung sinabi ni Dr. Paraan. Baka nga mayroon, baka may makita kapag finorgay natin yung signal na nakikita natin dito sa slide. Yeah. Baka lang, hindi ko alam kasi magkakaiba yung half width nila eh. So that's one. Let me continue yung question ko. Kasi gusto ko lang malaman yung number of fingers na sinasabi natin, it is integer, di ba? Tama ba ako?
Yes, opo. Yes.
Kasi doon sa movement, yung may slide ka, I failed to notice kung anong number. Yung nag-separate yung mga backing doon sa number of fingers. Mas marami, kung ba nagsama-sama, ano ba yan? Number of fingers, I think it's 20. Slide 20.
Dito po ba?
Yeah, doon sa slide 20, yes. Yeah.
20, slide 20.
20. Yeah. Doon sa walang backing, ito yung just your fabric, cotton fabric, floating.
Yes, opo. Yes.
So, may clear separation na sinasabi natin. Pero dahil hindi ko alam kung ano yung number dito, nakalog kasi yung y-axis natin. Hindi ko alam kung ano.
Yeah.
Although may separation nga, hindi ko alam kung integer yung tinitingnan ko. Kung
okay.
ilang number of fingers. But that's okay. Dito lang sa y-axis hindi ko kaagad nakita. Pwede mo lagyan na lang ng maliit na number diyan siguro.
Yeah. Okay, sir. I can show you, sir, the plot that is not logged.
Sige, pwede nga.
Check ko lang kung. Ayan, opo.
Ayun.
On the right side.
Okay. There.
Prepare ko lang.
Okay, very good. Then,
so you can see here actually that it approximately doubles. Yun yung sinasabi ko kanina.
Yes.
So, when you introduce a backing material from around 6, nagiging 12.
So yung number. Dun sa unback, dun sa just your fabric.
This one.
Up to
fabric.
Parang nag-saturate up until 5, depende sa velocity. Tama ako. Or height.
Yeah.
And then practically double to almost 10 or a little bit beyond 10 pagdating sa okay. Understandable yan. Pero yung separation ng dalawa, yung leather and aluminum, hindi natin ma-distinguish. Okay.
Opo.
Hirap na yun. Trinay namin yan before. Dropping a liquid on water, ink on water, and then watch the ink spread on water. Ganon din, practically ganyan din. Pag binago ko yung water and yung density ng ink and so on, properties ng ink, hindi namin ma-distinguish yung formation. So I think we're looking at it in the wrong way. But in this case, I think without and with backing, yun lang yung nakita natin. Tama ba ako?
Yes, opo.
By the way, yung fabric na stretch ba natin? Stretch or i-qualify lang natin na hindi sa stretch?
Loosely, ano ba yung term?
Spread.
Loosely placed on a, yung ginagamit sa, ano ba? Ganchillo ba yun? Yung hook?
Okay. Sige. I understand.
Opo.
Okay. I-note na lang natin dun sa manuscript din.
Okay.
Qualify.
Yeah. Yes, sir.
Kasi kung na-stretch din yung fabric, we might be seeing some yung dampening as you noted dun sa
Yes,
recommendation
apo.
mo,
Yes.
magkakaroon ng oscillation.
Yeah. Yes, exactly. Yes.
Kaya ko sinuggest yung volume na leading at saka front end. If you are going to introduce the oscillation, kung na yan iba na yung mechanics nung practically yung
Yeah.
pagbagsak, tapos ito gagalaw. Ganun yung nakikita. Yeah. Okay. Yung isang question ko nakita ko sa recommendation so sayang. Yung multiple droplets.
Yeah, opo.
Yung tanong ko kasi doon ay, have you considered not just the number, but actually the size? So, I'm thinking of the constant volume ng droplet is equal to the volume of all the satellites, including the mother pattern or the stain. Parang ganun. Kumbaga, kung habang lumalaki yung volume nung nalaglag, dumadami yung droplet, number lang yon. Pero hindi lang yung number, parang number times its size or may relation with its size para makuha natin kung constant. So one of the problem with this tracking ng droplets or satellites, kung mayroon tayong hindi na consider, malalaman natin. Actually, sometimes yung missing drop or missing portion gives us some information also. Parang ganun yung tinitingnan.
Yeah.
Leading yun kasi dun sa ownership nitong 2 droplets later or multiple droplets. Itong satellite, you might be able to identify which satellite belongs to which parent. Kapag mayroon tayong model ng constant volume or relation ng volume ng droplet in relation to the sizes, number and sizes of the satellite stains. Wala tayong nakita nun, diba? Wala tayong plot.
Well, is this the plot, sir, you're referring to? Sana?
Yeah, pero yeah. Okay, let me i-zoom ko. Ano yan? 38.
And I also have a plot of the so this is in relation to the different droplet volumes.
Okay.
With the number of satellites.
Yeah.
And I also have a plot of the area of the satellites or the size of the satellite with different droplet volumes as well.
Yeah.
So however, for the the sizes, walang separation or walang clear relationship. Yeah. And for the number of satellites, yun yung pinakita kong slide po kanina. So this is for the sizes of the satellites, so there's no distinction.
Okay. I see.
But for the number of satellites, but for the number of satellites, may clear distinction between the number of satellites and the droplet volume.
I see. Thank you. Di ko nakita yung y-axis nung isa. Area pala yun.
Yeah.
Okay.
Yeah.
Thank you, Wilbur. Wala na akong tanong. Thank you. Suggestion lang din. Maglagay tayo ng ranges of usability, basically parang range of volume, consider viscosity. Although we have that already, lagyan lang natin.
Yes, okay, sir.
Range of droplet speed. Hanggang dito lang yung itetest namin.
Yes, okay,
Ito lang yung i-qualify lang natin bakit dito sa range na to kayo tumingin. Yun lang.
Alright.
Thank
Okay.
you, Wilbur. Very congratulations sa nice presentation. Thank you.
Thank you, sir. Just a comment on Sir Sumintak's last suggestion. So this experiment is a passive draft experiment, and for blood stain pattern analysis researches, there is actually a distinction between the different types of droplets or stains produced by different staining methods. So for passive draft experiments, there's a certain range of droplet volume and impact velocities na ranges of values. So yun.
Yeah. Thank you, Wilbur.
Wilbur, ako rin may tanong.
Yes, ma'am.
Able to observe also blood stain beneath the in the backing material.
Anong slide po, ma'am?
Wala yata related lang dun sa backing material.
Okay.
I'm just curious if you were also able to, can you also see some blood stain in the backing material? Like
yeah.
you, for example, if you remove the
Fabric.
cloth, the fabric, yes.
Yeah. There is an observed staining on the backing materials as you remove the fabric when the experiment was done. However, we did not measure the characteristics of that. So maybe it can be a possible extension on how it relates to the interaction of the fabric with the backing material. But we can see a staining on the backing material for higher velocities. For lower velocities, I'm not really sure kung hindi ko masabi yung exact value, ma'am. But for lower velocities, the blood does not penetrate through the fabric. So walang staining. There's no staining at the backing material for lower velocities.
I was just wondering, maybe it can also help in discerning the impact velocity.
Mm-hmm.
The size of the stain beneath the fabric.
Yeah. Mm-hmm. Of course, that can be possible, and that is, I think, more appropriate if the backing material is not porous, so that there is no reduction in the volume of the droplet. One problem with investigating the stains on the backing material is that, for example, for silicon carbide, which is a very porous material, hindi na nade-discern yung stain. So nagsisip through kasi almost all the blood that interacts with the surface of the silicon carbide. So hindi siya, it's not clear, kumbaga. Pero yeah, that could be possible.
Porosity will also be a hindrance in calculating for example, if you want to know the total volume, you will use the conservation of the volume of the blood stains, like in
Yes.
Earlier.
Yes. Mm-hmm.
Why do you call it passive blood experiment or passive experiment setup?
Yeah, so it's called a passive drop experiment because the droplet is allowed to drop in the influence of gravity. So there's no the only external force that allows the droplet to fall is gravity. So kaya tinawag siyang passive drop.
In the projective transformation that you do on the raw image, is it because there is a deformation of the
No. Sorry. So in here, for the projective transformation was done mainly because the camera used was placed or was positioned at a glancing angle. as you can see here. That's why a projective transformation was done. The assumption here is since the droplets are vertically dropped from the in the fabric, there is a parang symmetry in the spreading. Does that answer your question, ma'am?
Do the other panel members have questions? Have further questions?
Dr. Bardolasa, may I ask a question? So this is passive drop. What's the opposite? Active? And what are examples of, say, an active drop?
So the other types of staining methods for in the blood stain pattern analysis are the ones I've mentioned in the introduction. So you can either have an impact spatter. This is produced when an object hits a pool of blood. And cast off. Another example is cast off patterns, which are produced when you swing an object and the blooded object ejects droplets that hit a certain surface. So for those cases, you have a higher impact velocity. Does that answer your question, ma'am?
Fair enough.
Are there other questions?
I have a question, ma'am. Jaira here. How did you make sure that the backing material is completely flat with the fabric?
So the before the dropping experiment, the fabric was placed. So ano ba i-term yun? So pinatong kumbaga, and then nag-apply ng force para lang dumikit dun sa surface, dun sa backing material. But if you are asking if meron bang mechanism so that it stays na nakadikit dun sa backing material, wala. So pinatong lang yung fabric, and then nag-apply lang ng force para lang lumapat ng maayos yung fabric dun sa backing material.
Okay, thank you.
Okay.
Any other questions from?
Wala na. Okay na ako kay Wilbur. Thank you.
I'm okay as well. Okay na ako.
Okay na rin po ako. Thank you, Wilbur.
Thank you, ma'am. Thank you, Dr. Samintak. Thank you, Dr. Paraan and Dr. Soriano.
Before we let you go, it is required that we take a picture together with the panel members as proof of your defense. So if I may ask the panel members to open their video. I'll remove my mask first. Okay. So there's Dr. Paraan. So, okay, I'll take a screenshot. Screen 1, 2, 3. Got it. Just a moment. And then, just a moment, please. Isi-save ko lang. And then with the rest of the guests, kung pwede, this is not required, but at least those who are here, you can stop sharing your slide,
Okay.
Wilbur, so that we can take a picture of your guests. So welcome guests to open your video so that we have some memory of Wilbur's defense and your attendance. Thank you very much. Printing screen. Yes. That too. So please go ahead and take screenshots as well for your purpose. Okay, got it. Okay.
Thank you
very much.
Thank you, friends.
Okay, I will end the YouTube streaming so that the panel can discuss. Okay, and then the rest of the guests can